afternoon everyone, this is Zafar from Ahmadiyya International School. Welcome to this online classes from Ahmadiyya International School. I have selected a few important topics for revision for you people. What you can do is you can watch these classes, and if you have any questions or any queries, you can audio record your messages and send it to me on Viber. So, if your questions are good, those questions will be selected for panel discussion that will be coming up next. So, watch them, and if you have any questions or any queries or any explanations needed, you can audio record a message and send it to me on Viber. Remember that, okay? So, the important topics that we have chosen for these discussions are, the first topic is about protein synthesis. We will discuss how proteins are made. So, this will be divided into two classes, the first class being transcription. The next class will have part 2, they will be discussing about translation, the next part of it. So, the topic is protein synthesis, and in this topic I will be discussing about transcription only. In the next class we will be discussing about translation. So, first thing, what is protein synthesis? Protein synthesis is nothing but how proteins are made in the cell. Now, you might say, why proteins? What is the importance of proteins? Now, if you look at the proteins in our cells, there are about I would say 90,000 different types of proteins, more than that, more than 90,000 different proteins are there. But if you look at a cell, now if I look at a cell, there are so many types of organic compounds within a cell. Carbohydrates, you have lipids, you have proteins, you have vitamins, but out of all of them, the most important, by far the most important ones are the proteins. So, they play a very big role. They give you the characteristics, the way you look and other things. Now, first I'll talk about the importance of proteins. Why proteins? The first thing, proteins are useful for growth. Proteins are, all your enzymes are proteins, the hormones are proteins, your pigments like hemoglobin is a protein, keratin is a protein, you have this collagen, they have varied functions. So, proteins are the most important molecules present in a cell, and we are going to discuss about how these proteins are made within a cell. So, this protein synthesis is divided into two stages. One is called the transcription, the first stage, and the next stage is called as translation. So, let us learn the definitions first. What do you mean by transcription and what do you mean by translation? Transcription is a process by which information on the DNA, shortly we will be discussing what is this DNA. So, the information on the DNA is copied into a messenger RNA. Copied into a messenger RNA, that is called as transcription, and this happens inside the nucleus. So, here DNA information copied into a messenger RNA. The next stage, which is called translation, translation takes place in the cytoplasm, where mRNA information is used to attach the amino acids to form a protein. So, this is the procedure, this is the process where the proteins are made, this is the process where mRNA is made. So, there are two clear cut, be very clear with this, what is transcription and what is translation. Transcription happens in the nucleus and translation takes place in the cytoplasm. Before we go further, let us discuss about the structure of DNA. It is very important that we learn the structure of DNA first. And DNA is nothing but deoxyribonucleic acid. You know, it is a nucleic acid present in the nucleus, and it is a polymer. It is a very long polymer made up of nucleotides. So, DNA is a polymer made up of nucleotides. Nucleotides are the monomers, like the bricks that make up the wall. So, if I talk about one nucleotide, it has three components, three parts in a nucleotide. You have this pentose sugar first. In this case, it is a deoxyribose sugar. Attached to this one, you have a nitrogenous base to the first carbon. Then you have the phosphate group attached to the fifth carbon here. So, this is one nucleotide. Remember, the basic component or the monomer of DNA or the RNA is a nucleotide, and which itself has three components. The components being the pentose sugar, I am talking about any type of nucleic acid, pentasugar, then you have the nitrogenous base, and then you have the phosphate group. These three together forms a nucleotide. So, this is the basic unit. Now, let us discuss about the pentasugar. The pentasugar, you know very well, it is a deoxyribose sugar in the case of DNA, but if it is an RNA, you have ribose sugar. So, this is a deoxyribose sugar. Attached to this one, there is a nitrogenous base. Now, it is very important, you should know all the nitrogenous bases. So, this nitrogenous bases, if I talk little bit about the nitrogenous bases, the ones which are found inside the DNA, you have adenine, thymine, guanine, and the cytosine, no uracil. If it is RNA, you have uracil instead of the thymine there. So, you have the nitrogenous bases adenine, thymine, guanine, and cytosine attached here. Okay? And name the bond here, this is the glycosidic bond, or you can say N-glycosidic bond between the pentose sugar and the nitrogenous base, and this is the phosphate group. So, this is attached to the 5th carbon here, this is a phosphoester bond, and this whole thing is called one nucleotide. And so, I think it is clear what is the structure of a nucleotide. Now, we will learn about how two nucleotides join together. So, this is one nucleotide. I told you DNA is a polymer. So, this is one nucleotide, this is another nucleotide, and listen carefully, between these two nucleotides you have a bond here. You should know the bond here. This bond is the phosphodiester bond. So, they will ask you a question, name the bond present between two nucleotides. The bond present between two nucleotides is the phosphodiester bond. So, here also you have a phosphodiester bond. Like this, so many of them join together and they form a long strand like this, long strand, and this line represents the sugar phosphate backbone. Sugar phosphate like this. I will just draw it like a strand, and the nitrogenous bases will be hanging out or projecting out like this. This is how we draw it. This is one strand of DNA. On the other, you know very well, DNA is a double helix which is made up of two strands. So, there is another strand here. I will draw it on the other side here, and this will be like flipped or inverted like this, and you have the nitrogenous bases here. So, why is it flipped? So that it can react with this nitrogenous base. And I told you the nitrogenous bases in a DNA or adenine or thymine or guanine or the cytosine. So, adenine, here it will be a thymine, another nucleotide, deoxyribose nucleotide, let us say C here, G here, and you have one more here, let us say it is like this. Now, you see carefully, between these two nitrogenous bases you have hydrogen bonds here. You have the hydrogen bonds. How many bonds here? 2. Remember, it is very, very important you learn this. Adenine always binds with thymine by 2 hydrogen bonds. Cytosine always binds with guanine by 3 hydrogen bonds. You might ask, is it must? Yes. All the organisms, they follow this rule, and this rule is called as complementary base pairing rule. Remember that always. Complementary base pairing rule states that adenine always binds with thymine in the case of DNA with 2 hydrogen bonds, and cytosine always binds with guanine by 3 hydrogen bonds, and that keeps the 2 strands together. These hydrogen bonds, they keep the strands together when it is folded into a double helix. So, you can see here, and this one is folded into a double helix like this, and that is the structure of a DNA molecule. So, I think here you know how very well. Now, do you agree, if I know one of the nitrogen bases on one strand, I can guess the nitrogen base on the opposite strand, because I know it is a complementary base pairing rule. If you tell me on one of the strands it is guanine, I know on the other strand it is cytosine. Okay? So, we know very well, if I know one of the nitrogenous bases on one strand, I can guess the nitrogen base on the opposite strand, because they always follow the complementary base pairing rule. Okay, I think the DNA structure is clear now. Okay. Now, let us discuss about gene now. So, you might say, sir, we learned the DNA now, and what is a gene by the way? So if I talk about the DNA double helix, you know very well, it's a very, very long structure or a polymer having a double helix like this, very long, very, very long, thousands and millions of nucleotides joined together. It's very, very long. I'm just drawing a part of it. Now in this long DNA, you have coding areas. Maybe from here to here, this part is a coding part. What do you mean by coding part? Coding part means this part of the DNA helps to make a protein, and that part is called as a gene. So we say genes, he has the genes for blue eyes, he has the genes for being tall. So what do I mean by gene is nothing but it's a part of the DNA that helps to make a protein. I'm not saying it will completely make a protein, I'm saying it helps to make a protein. Okay? It has the information about how to join the amino acids together. Again, you have a waste part, or I should say the non-coding part. Non-coding part, it continues, let's say from here. I'm just randomly choosing some places here. This will be another gene, maybe gene 2. Then you have the non-coding part. These non-coding parts are called as introns, or you can say the junk DNA. And the coding parts are called as the genes. So I think it's very much clear now what are genes. So if you can imagine like this, you have a long white thread, a very long white thread, and in that white thread you have colored areas. So the small colored areas will be the genes, and the white parts will be your introns, the non-coding parts, which don't help in protein synthesis. So what are genes? Genes are the parts that help in protein synthesis. They help to arrange the amino acids together. So I think this part is clear. Now the next thing that you might ask me is, what about the length of a gene? And is it same for all the genes? No. Some genes are very short, some genes are very, very long, sometimes millions of nucleotides long, sometimes it's very, very short as well. And how many genes do we have? Human being cells, we all have around about 25,000 genes, 23,000 to 25,000 genes in each and every cell of your body. Not in the whole body, in each and every cell you have about 23,000 genes which will help in protein synthesis. Before we go to the next part, let us discuss a little bit about the amino acids, because it will be helpful later. So let us discuss about amino acids. What are these amino acids? Because we learned that in protein synthesis, amino acids are joined together to form the protein. These are the monomers that will give rise to the polymers, right? So what are these amino acids? Let us discuss about the chemical structure first. So they have a central carbon, they have an amine group, they have a carboxylic acid group, hydrogen here, look at this, central carbon, amine group, not amide group, amine group, NH2, then you have carboxylic acid group here, that's why they are called as amino acids, amine group and the acid group is there, you have a hydrogen on the other end, and this side you have the R group. Now this R group is very much variable. What do you mean by variable? It may have different elements there. It may have sulfur, it may have iron, it may have a ring structure, it may have a long hydrocarbon chain there. So how many types of amino acids we have? We have 20 different types of amino acids. 20 different types of amino acids. For example, leucine, alanine, valine, so many are there. Should you remember all these names? No, at least know one or two names. So you have these names like leucine, alanine, valine, so many of them are there, 20 different types are there. Now if you ask me what's the difference between these 20 amino acids, the difference between these 20 amino acids is the R group. This is the functional group of an amino acids. So the amino acid might be polar or non-polar, or it might be soluble or insoluble, it might be charged or uncharged. All these properties depends on the R group. So you have basically 20 different types of amino acids, and all these amino acids, they may have different properties based on the R groups. So in a cell you have so many types of amino acids in the cytoplasm. Now the next question that comes is, how did these amino acids come in the cytoplasm? Can our cell make some amino acids? Yes, our cells can make some amino acids, and those amino acids are called as non-essential amino acids. That means you need not consume them, your body can make them, okay? But there are some amino acids which cannot be synthesized by your cells and you have to consume them. You have to eat them. I mean, you have to eat them in the sense, people who are eating only one type of food, let's say every day they are eating the same type of food, they may have deficiency of some amino acids in their body, because all the foods do not contain all the amino acids in them. Remember that, please. So it's better to have a variety of diet, because there are different types of amino acids present in different types of diets. Okay. So I think it's clear. So amino acids are there, you know the structure very well. Now let us discuss about how they join together. One amino, it's very easy, because it helps us to understand protein synthesis. So how does one amino acid join with another amino acid? So here you draw another amino acid here. The amine group, the carboxylic acid group, the hydrogen and the R group. So you can see here the hydrogen of this group, the amine group of one amino acid and the hydroxyl group of the carboxylic acid, they are removed by condensation reaction, removal of water molecule, and they form a bond here. And this bond is called as a peptide bond. I repeat again, the amine group of one amino acid and the hydroxyl group of the next amino acid, they join together by forming a bond, and that bond is called peptide bond. And remember, it's a condensation reaction. And what's a condensation reaction? A condensation reaction is one in which water molecule is formed. So that's how amino acids get attached to one another: valine, leucine, alanine, histidine. So this is the peptide bond, peptide bond, peptide bond, and condensation reaction. Ultimately, you get a polypeptide or a protein. That is how amino acids join together. The next thing that the students should remember is, sometimes they get confused between nitrogenous bases and amino acids. Really, they do get confused between nitrogenous bases. So there is no need of confusion here. Nitrogen bases are a part of the nucleotides. The nitrogen bases are adenine, thymine, guanine and cytosine, as well as uracil. Amino acids are the building blocks for proteins. So please don't get confused between the nitrogenous bases and the amino acids, which are completely different. The next thing, very important thing now, we know very well, coming to protein synthesis now, you know very well that the DNA is in the nucleus here, and the amino acids are in the cytoplasm. They came from the food that you ate. The proteins got digested, and here you have the different types of different types, thousands and thousands of amino acids. And I told you, transcription is a process by which information on the DNA is copied into an mRNA, then that mRNA is used to attach these amino acids together. Now look at this, we have a problem here. Amino acids are in the cytoplasm, the gene part is inside the nucleus. Now what do you expect? If I draw a little longer one, generally you might think like this DNA or the gene part would come out of the nucleus, DNA coming out of the nucleus, helping to attach the amino acids, help to attach the amino acids and go back. Isn't it? You may feel like, you think like, okay fine, DNA has the information to attach them, so the DNA will come out. Now this doesn't happen, this is too risky, or you can say this is not good actually. If the DNA comes out of the nucleus, there are two problems here. One thing is, it might be attacked by enzymes, or it might be lost in the vast cytoplasm. It may not find its way back. So the DNA never comes out of the nucleus. Instead, it makes a copy of the DNA, which is mRNA. This mRNA comes out into the cytoplasm. To make you understand much better, I can give an example. For example, if you are making a house or a construction is going on, so you will have an original copy, that's called as a blueprint. So if you have a blueprint, if I give a blueprint to an employee or maybe a worker, he may lose that blueprint. So I may not have any other copy of that one. So it's gone forever. But instead what I do is, if I take a blueprint and if I take a photocopy of that blueprint and give it to a worker, even if he loses it, the original is safe. The same thing here, the original is safe, the information how to make a protein is safe inside the nucleus. The DNA makes a copy which is called as mRNA, and this mRNA comes into the cytoplasm to attach the amino acids together. So that procedure where the information of the DNA is copied into an mRNA is called as transcription. Now, let us go into the details of how transcription takes place. Okay, now let us discuss about transcription now, how the transcription takes place. You know very well, we already discussed this, DNA is very, very long and you have these coding areas which are called as genes. Now, there is an enzyme inside the nucleus which is called as RNA polymerase, not the DNA polymerase. You have this RNA polymerase which has to open up this part of the DNA and copy this information into an mRNA. So, remember one thing, I am not going to copy the whole DNA from the beginning up to the end. I want only the gene part to be copied. It is very important. Remember that. I want only the gene part to be copied. So, how would this enzyme come to know that the gene is starting from this particular point onwards? So, that is because of an area called as a promoter region. So, just in front of a gene you have a promoter region that tells this enzyme called RNA polymerase to come and bind here and start the process of transcription from here onwards. Do not copy from here, it is very important. If it copies from here, the whole protein made will be a wrong one. So, that area is called as a promoter region and this enzyme binds to the promoter region along with some transcription factors. And once it binds, it opens up this DNA double helix, copies this information, and at the end of a gene there will be a stop signal as well that tells the enzyme to not to copy the information from this point onwards. So, I think the part is clear. So, you have a promoter and then you have a terminator sequence, or you can say the stop signal for the RNA polymerase. So, again here this enzyme called RNA, if you look at this picture here, the RNA polymerase unwinds the DNA double helix by breaking down the hydrogen bonds between the two helices or between the two strands. And remember, for transcription only one strand is used, not both the strands, unlike DNA replication. In DNA replication both strands act as template, but here only one strand acts. That strand is called as a template strand, or you can say, it is a very important point, you can, it is a one marking point if you write this, it is also called antisense strand. So, I think it is clear, out of the two strands of a DNA double helix only one strand is used and that strand is called as a template strand. So, this is being used in this case. So, this is, and remember one thing, the whole DNA is not unbound, only the gene part is unbound, this is as it is intact, this might be the promoter region here. So, this is the one that is opened up. And these guys, or you can say these ribose mononucleotides from the nucleoplasm, they are taken or they will align themselves near the complementary base pairs. You know already what are complementary base pairing, cytosine always binds with guanine. So, a nucleotide which is having cytosine aligns itself opposite of cytosine here, this is having guanine. So, they form hydrogen bonds with the help of the enzyme, the RNA polymerase. So, the complementary base pairs align nearby the antisense strand and this enzyme attaches them by hydrogen bond. You should write just, you just can't write just they attach, you have to write they form the hydrogen bonds. Is it clear? So, let us say that it has formed hydrogen bonds here, you know very well 3 hydrogen bonds here, 2 hydrogen bonds here, 3 hydrogen bonds between cytosine and guanine. And remember, these two, the ribose mononucleotides, they should get connected, and they get connected by phosphodiester bonds by condensation reaction. There are 2 marking points here. One is you are naming the bond, the other one is for writing the name of the reaction. It is a condensation reaction and the name of the bond is phosphodiester bond. Here you have another phosphodiester bond. Like this, one by one, bits and pieces get this nucleotides which are single pieces, they get attached to the antisense strand. Now, what am I getting here? I am getting a long strand of mRNA. This is the mRNA. These are temporary hydrogen bonds, remember. These temporary hydrogen bonds are broken one by one, and this mRNA comes out of the DNA double helix. So, I think this part is clear. If they ask you for 4 mark, because this question always comes for 4 mark as an asterisk question where you have to write the in a logical sequence, write the technical terms correctly. So how are you going to write this answer? First point, you are going to write enzyme called RNA polymerase binds to the promoter region and starts unwinding the DNA. Only one strand of DNA double helix is used, and that strand is called as a template strand. Free ribose mononucleotides from the nucleoplasm, they align themselves nearby the complementary base pairs. Enzyme called RNA polymerase attaches them by following the complementary base pairing rule. Cytosine binds with guanine by 3 hydrogen bonds, uracil binds with adenine by 2 hydrogen bonds, remember that. And then next marking point will be 2 ribose mononucleotides are connected by phosphodiester bonds by condensation reaction by removal of water. And this process continues until the stop signal is reached. Once the stop signal is reached, RNA polymerase leaves this place after breaking down these bonds. This mRNA comes out of the double helix, okay? And this becomes again, once this is gone, this becomes again, they form the complementary base pairs, they form the hydrogen bonds, again it becomes a double helix. So if I have to show you with my hands, actually this is a double helix. It's unbound, one strand is used, mononucleotides are attached one by one by following the complementary base pairing rule, and they get connected to one another. You get a mRNA here, so this is another strand, the sense strand, and this mRNA comes out of the nucleus, and that goes for the next stage, that is called as the translation. Hope it's very much clear. If you have any doubts, if you need more explanation for something, please send a message and we'll be discussing your questions in the panel. Thank you very much.